Good morning, everybody. For those of you who didn't meet me yesterday, I'm Day Behbehani. I'm one of the clinical research postdocs in the Nolan lab. And I'm going to talk to you today about the methods that I developed for doing cell cycle analysis. This relates a little bit to the stuff that Harris is going to tell you next. I'm also going to touch briefly on spade analysis, which some of you may starting to become familiar with, but you're going to get a much more detailed lecture on that later today. So today, as I said, I'm talking about the application of mass cytometry to looking at proliferation during both normal and malignant hematopoiesis. I won't really get too into the malignant things just a little bit at the end. But in general, I'm going to talk to you about the basic method for cell cycle analysis, the markers that we use to mark cells in S phase, G0, G1, G2, and M, and the basic validation experiments that we've done with cycling T cells. And then I'm going to go on and show you how you can then fold this into a much higher system level analysis of cell cycle using spade clustering and looking at some data that we've published looking at the normal bone marrow cell cycle state and comparing that to some malignant samples. So all of the normal cell cycle stuff was published recently in Cytometry, and you can go get that July issue if you want to see all this in detail. Almost all the figures I'm showing will come from that paper, so it's a great reference if you want to try and actually repeat some of this. So you all may be aware that there is what we call a DNA stain for the cytoph. It's this iridium intercalator. This is what it looks like chemically, but you might think that you could get cell cycle information from that, but unfortunately you cannot. So here we see the typical host stain distribution that you see with this typical G1 peak, S phase region here, and G2 by DNA content. This is not sort of optimal for resolving the cell cycle, but you can get a pretty good guess of S or G2, or G1, S, and G2 phases. It doesn't really tell you much about M phase, but by contrast, if you take this iridium intercalator and stain it under appropriate conditions, you get a curve that looks something like this. Now, as it turns out, this does actually correlate with cell cycle. You can see that the G0 cells here have less than the G1 and the S or the G2, but there's no way you can really gate on those populations. So we needed a new methodology if we wanted to do cell cycle analysis by cytoph, and my interest is in how cells respond to chemotherapy. And so I knew that we would really need some way of looking at cell cycle to truly interpret that. So what we started with is using iododeoxyuridine. Iododeoxyuridine is just essentially very similar to bromodeoxyuridine. In fact, in some of the earlier papers, people started working with IDU before BRDU. It functions essentially the same in sort of standard assays. In fact, you can use a lot of the same antibodies to detect IDU as you do BRDU and use the exact same procedures. But since it has iodine, and iodine has a molecular mass of 127, we can actually detect it directly by cytoph, which was actually a little surprising in the sense that we are getting a positive iodine atom that we detect, but it does seem to work just fine. So if we put this on the cells, we can rapidly detect it in the DNA of the S phase cells. Again, the beauty of the cytoph is that we don't have to use an antibody to detect this. It gets incorporated in the DNA, then we vaporize the cell, and we vaporize the DNA with it, and then we see the iodine coming out directly. And that gives it very high sensitivity. You can see that we can basically define, for this population of cells, it has about 23 to 24% S phase cells. Within 5 minutes, we can identify that population. And our typical experiments are done at 10 to 15 minutes. As you can see, the longer you go, the more iodine is incorporated into the S phase cells. The fraction by 60 minutes does go up slightly, but there are some cells that are entering S phase during the 60 minutes, so you would expect that to slightly go up. And so really gives quite clean S phase data. This works at the concentrations that you typically would use iododeoxyuridine. In fact, you can probably cut it even lower than the typical 10 micromolar that you see here. And thus we're able to define quite readily the S phase cells. And this sort of remains the easiest way of of doing cell cycles just to get a simple S-phase analysis. The other steps are a little bit more complicated. They're still based on this because we need the S-phase to kind of orient all the other cell cycle analyses, but just doing S-phase analysis with IDU is really straightforward. A number of other people in the lab have gotten this to work quite readily, and we've gotten some collaborators using this, and a group in Toronto has also somewhat independently come up with the same approach. So this is really straightforward if you want to try and do it. You just get the IDU, add it to your cells, and you can get the S-phase fraction quite readily. But we wanted to get at the other phases of the cell cycle as well, to ask questions about, are cells quiescent? Are they arrested? How might this impact whether they respond to chemotherapy? So we needed other markers. And so you can see in this schematic of sort of the basic cell cycle, just about everything here is potentially a target. So we have antibodies to phosphorylated RB. You can get cyclin and CDK antibodies, as well as antibodies to phosphorylated histone H3. And so we explored a variety of these targets to see which of these might function well to define the other phases of the cell cycle. And the ones that we found most useful were cyclin B1, phosphorylated RB. Now this is a slightly different epitope than the 788 that most people are familiar with. This is 807, 811, that in a Cell paper from about 5 or 6 years ago was shown to correlate with the G0, G1 transition. And then phosphorylated histone H3, here we're using the S28. S10 also works. And this is a well-established marker of M-phase cells, and it's quite bright on those cells that readily defines that population. And so by using this, you can define a population that is using an IDU by cyclin axis here. You can define a population that doesn't have IDU incorporation and is low in cyclin B, which contains the G0, G1 cells. Obviously, the S-phase population we can define easily just based on the IDU incorporation. And then the cells that have high cyclin B1 but are not in S-phase, we can find are the G2 cells. Then for phosphorylated RB, any cell that has as much phosphorylated RB as an S-phase cell, we can define as a cycling cell. And those cells that have less than an S-phase cell are the G0 cells. And then just like with typical analysis, the phosphorylated histone H3, S28, clearly defines an M-phase population. And so with this approach, you can essentially define all the major phases of the cell cycle. And so you can take the same cells and define these markers both fluorescently and using mass cytometry. Here we're seeing sort of the mass cytometry plots. Again, phospho-RB versus IDU to define your S-phase cycling cells and your G0 cells, and then IDU by cyclin B to define G1, S, and G2. You can use these same plots with fluorescent antibodies against the same targets. If you, of course, have to use an antibody to detect the IDU incorporation, but analyzing these same cells by fluorescence, you can define these same populations. And this may, in some cases, be a good fluorescent approach, depending on what channels your antibodies might be on. When you're doing a host or PI cell cycle distribution, you have to use a ton of that to fully intercalate the DNA, and that can really spill over into a lot of other channels on fluorescence. So it's possible for some applications, just fluorescent applications, you may want to use this technique to define your cell populations, particularly if there's only a couple phases of the cell cycle that are really important to you. So by doing this, we were able to show fairly close correlation between the cell cycle distribution of basal cells by both fluorescence and mass cytometry. Here we did this in replicates of, I think, 5 cells, and you can see pretty close correlation. A little bit of disparity in the G0 and G2 compartments. Some of this is due to, particularly in the G0 compartment, due to fragments of cells that we can't, without scatter, have a hard time gating out. So there's always a little bit more cell fragment noise with the mass cytometer, so that can be an issue, and those fragments tend to look like G0 cells because they don't really have enough of the markers to be positive for other things. But pretty good estimation of the cell cycle distribution. To validate this, just make sure we weren't looking at some coincidentally correlated artifact. We took resting T cells and stimulated them with PMA and inomycin into cycle. and then compared the same cells by both the fluorescent approach and the mass cytometry approach, you can see at time 0 we'd expect all the cells to be in G0, and indeed they're all falling in the G1, G0 gate. And then on the Hoesch by pyronin fluorescent analysis, they're all here in G0, so G1 content of DNA and pyronin low. Looking by mass cytometry, again, the same thing. They're falling in the G1, G0 gate, and they're all lower in phosphorylated RB than the rare S-phase cells that sneak through. By 28 hours, you can see that the cells in both cases have started to intercycle. They're starting to incorporate IDU. You can see a big shift from cells in terms of their pyronin positivity. And when we look by mass cytometry, we see the same thing. They're starting to incorporate IDU, and we see a big shift in the phosphorylated RB in response to entering cell cycle. And by 48 hours, we see sort of a complete cell cycle distribution in both cases. These are stimulated T cells, so some of them aren't very happy and sort of stay in G0, but that's similar with both techniques. And so just sort of plotting out the relative percentages, you can see that they're quite similar. And of course, we're getting an orderly accumulation of the cell cycle state, so it's not some sort of artifact clearly in cells that we know where they should be. They are where we think they are by both techniques. So now we've essentially defined how to determine the cell cycle state with essentially as few as 4 markers, one of which actually doesn't use up an antibody channel. And so that leaves you a lot of other markers to do whatever other biology you might be interested in. And so as a first sort of proof of principle, we took normal bone marrow, treated it with IDU, fixed it fresh, and then analyzed the cell cycle state of all the different immunophenotypic populations that we could identify. So we used spade clustering to visualize that data, and that was part of the cytometry paper that you can look at. And then once you've developed this, we can do comparisons of sort of normal cells to malignant cells and look at how that might correlate to characteristics of the patients. And I'll just touch on that briefly at the end. So looking at the data is somewhat difficult when you get this much data. And so if you do a simple 4-parameter experiment, there's really only 6 biaxial plots that are generated from that data. It's pretty easy to look at those and get a sense of what your data looks like. You do a fancier fluorescent experiment with 8 parameters. Well, now you've actually got 28 different plots that you could look at, so that gets a little bit complicated. If you're really great at fluorescent flow cytometry, you can maybe do a 14-parameter experiment, and that will give you 91 possible biaxial plots, which is starting to get pretty daunting. But let's say you do a CyTOF experiment with 32 parameters, and the panel I'm currently designing has 39, so this only gets worse. But you can get a pretty staggering number of biaxial plots. So obviously, this is not an easy way to look at your data as a whole. And that's what necessitated the development of SPADE. This was developed primarily by Peng Kui in Sylvia Pulveridis' lab, but we've obviously taken it and applied it to the CyTOF data with a lot of success. I'm just going to talk to you very briefly about what SPADE is. Again, you're going to have a whole lecture on this, and some of you may have already get some sense, but just so that you can interpret the data I'm about to show you. The basic idea here, sort of visualizing 3 dimensions of data, is that you have a cluster of data in 3 parameters, and you can see there's some sort of dense regions of the data and some really sparse regions of the data. And the first step of this to get good representation is to downsample the dense regions. So that's what the density-dependent downsampling step is. You can see that after that step, we've gotten rid of a lot of the cell events in the dense regions and sort of evened out the distribution of these cells. That's important just so that regions don't just get ignored during the clustering step. From there, we just do n-dimensional clustering. So here we're showing 3 dimensions because it's easy to visualize, but we're typically doing this in about 15 or so dimensions of the data. It does get a little noisy when you try and start doing 30-dimensional clustering. It also gets quite computationally intensive. So the sweet spot is somewhere in the I'd say 7 to 15 range of parameters that you cluster on. But obviously that gives you a lot of diversity. And you can see that this clustering step forms multiple different clusters in the data. And then to make it visualizable, we do create a minimum spanning tree that basically says how related are these clusters to each other, and then attaches them by their degree of relatedness. In this plot, the length of the line that connects the two clusters is not meaningful, and the shape of the tree is not meaningful. In fact, we manipulate it just to make it easier to draw boxes around things and make it easier to present the data. But what is important is the relationship. So what cluster connects to what other cluster is the key piece of information that this is showing you, and that we don't ever change. So ultimately, you get a tree structure like this that we sort of bend into a pleasing configuration that helps us show whatever we're trying to show. But the relationships between the nodes are maintained and based on the spanning tree. From there, of course, the software has no idea what a T cell is or what a B cell is. It just says these are clusters of cells that are closely related by however many dimensions you told it to cluster on. So what we then ask it to do is show us what the median of each cluster is for specific markers. And so it will say for CD3, we have a cluster that has really high CD3 and then basically low or minimal CD3 everywhere else. Then we can say, well, what about CD8? You can see that one arm of that cluster is CD8 positive, one arm is CD4 positive. Same thing works for the other things. You get like a B cell arm that's indicated by 19, and some of those are immature. They have only 19 and no 20. And then you can cluster on other things that are sort of distributed evenly, like CD38. They can help you define early progenitor cells and other similar populations. So at the end of the day, based on our assessment of what these patterns look like, what these expression patterns look like, we then assign these cells sort of to correlate with standard gates. Again, we're making these assignments based on the median levels of expression in these nodes. The software doesn't do this part. So we manually annotate this to show you what cells are in which cluster. And now this becomes a really useful way because we can color the tree for whatever parameter we're interested in and show you what's the expression of this, that, or the other across all these different cell types, and you can see it all at once. So we did that here for a normal bone marrow sample. And you can see we were able to define basically all the different immunophenotypic populations that you might expect, from HSCs all the way out to CD8 naive and resting T cells, all the different steps in erythroid, major steps in erythroid development, granulocytic development, B cell development, and so on. And we ran this, again, on cells that had incorporated IDU that we could then do cell cycle analysis on. And so here I'll just show you the component coloring that might be relevant. So you can see the T cells are positive for CD3. The granulocyte lineage is positive for CD15. The monocyte lineage is positive for 14. CD20 is in the B cells. CD235 is in the erythroid cells. And the progenitors have CD34 like you'd expect. So let's go one arm at a time. We'll start with the B cell arm. You can see that using a combination of markers, we were able to define all the different B cell steps. So a high CD34 high pro B cell population that had really no significant 19 or 20. Then a pre B cell population that has a little bit of 34, a fair bit of 10, and a tiny bit of 19. A pre B2 that now has 19 but no 20, and then a mature population that has both 19 and 20, has no 10 or 34. From there, we could figure out what the cell cycle distribution was in each of those populations, and I'm showing you the cell cycle distribution here. These sort of circle and circle plots, essentially just for ease of visualization. The size of the red circle indicates the fraction of cells in that phase of the cell cycle. Just to make it easier to visualize, a completely full circle is only 67%. You can set this up however you want, but if you make it to a full circle 100%, these start to get kind of small. But you can see that most of the G1 cells and the S phase cells are concentrated in this pre-B2 population, which is also much larger than the earlier pro-B and pre-B1 populations, as you might expect, and so that the majority of proliferation of B cell development is coming from this pre-B2 population. We can do the same thing with the erythroid. Sorry.
Can you flip back?
Sure.
So what are these populations gated on? C34, C10?
Yeah. I mean, essentially, I color the tree just as you see here. And I use that information. I mean, I can pull up the biaxials for any of these groups, but I use that information to determine which cells are which. The clustering's not perfect, so one could argue, well, is every one of these cells really a pre-B1? There's some here that don't have a lot of 10. And, you can make those arguments, and it's probably true. But it's really, you can obviously go back to the data and manually gate it, and then look at it and show biaxial plots, and I have all those. So while not maybe only 98% of what I'm showing you here and calling a pre-B1 probably fits most people's criteria, but it's a much easier way to sort of look at this amount of data.
And what's the rationale? What's a good logical criteria to gate these cells?
What's a good logical criteria to gate them by regular fluorescence? You do lose scatter, and that can be important actually. A lot of the traditional gating strategies are really quite dependent on scatter. But actually, when you have as many markers as we have, you can find lots of other things that sort of correlate. 45 is really useful. We can get a little bit of cell size information, but we've sort of had to take what we know and make our best estimations. From work like Sean and Aaron's work using phosphoflow from the science paper, it also helps us know, here are cells that signal clearly like this population, and then I can go back and say, well, what were the immunophenotypes of those cells? And then I can use that information to help me make my gates. And so it's a combination of all of those things that have sort of helped us derive this. But the science paper is a great reference. In the supplemental information, there's a table that describes why do we call everything that we called what we called it. Mostly that's immunophenotypic information, but some of it was signaling state. Some of it's just that this population here is clearly this population here is clearly that. The only thing in between is exactly what we'd expect to be in between, but we didn't really have a great marker for it, but everything that we did have was consistent with that, and so that's our best guess. Looking at the erythroid population, we can do the same thing. Again, you get like your MEPs and pro-erythroblasts that have 34. Then you have erythroblast populations that have high CD71 but don't have 34. And have sort of intermediate CD235, and then you have populations here that are high in 235 that are becoming low in CD71. They're sort of your more mature nucleated red blood cells. Again, you can see that almost all the proliferation is taking place within that erythroblast population, just like you'd expect. Though in the pro-erythroblast, even though they're small, you can see that some of those proliferate pretty well. There's very few MEPs, so it's hard to say, but I think the actual proliferative rate was like 30% in those, something like that. And I think I mentioned to some of you guys yesterday in my group that G2 populations in these in vivo assays tend to be quite small. And if you look at other publications of normal cells like ES cells or in vitro derived cells where cell cycle analysis is done, again, they see the same thing, that there is actually the G2 population is quite small, much smaller than what we see in cultured cancer cell lines, where I believe probably most of the G2 cells you're seeing are actually G2M arrested cells. So similarly, we can get the same data from the myelocyte lineage. And again, we see most of the proliferation coming from the early and late pro-myelocyte populations. So having done that, we then started applying this to both normal and malignant samples. And here I'm just going to show you about two sort of pilot experiments where we looked at AML samples using this approach. Again, here's the tree that we were able to derive, and basically the same approach that we had taken last time. And by doing that, we could define two different arms where most of the AML cells, blast cells, were contained. So in this sample of 5, you can see most of them were sort of in this arm, but we also saw some AML cells clustering into some of the more mature populations. In AML 9, most of the cells were actually in this arm, with some clustering into some of these more matures. They had slightly different CD34 expressions. And you can see that if you look at their cell cycle distributions, they were markedly different. So you're seeing different immunophenotypes. You see different relative amounts of proliferation across different developmental stages. In both of them, you can see some that are too immature to really have much proliferation and too mature to have much proliferation. And you can see definitely different patterns in the different patients. And our hypothesis is these patterns are going to correlate with these patients' outcomes, and that's something we're sort of actively working on now.
Can you go back to the T-cell branch?
The T-cell branch of this tree?
No.
So we didn't really look, I didn't show you the cell cycle distribution of the T-cell branch here, because essentially they're not dividing.
The cell cycle distribution, but the naive T-cells, the naive CD4 T-cells, it look, maybe I just don't understand how it's arranged. It almost looks like the naive CD8 T-cells are a sub-population of the naive CD4 T-cells. Is that line that's connecting them, a hierarchical line?
So this is a common misconception about SPADE. So you know the SPADE plots that we did in the original science paper, they just so worked out to mirror development that everyone sort of thinks that it tells you something about development. It doesn't. It just tells you about relatedness. And surprisingly, often relatedness in terms of immunophenotype clustering does follow development, but not always. There's no strict rule, and I wouldn't ascribe any meaning to which things are next to each other other than that they are immunophenotypically similar within the markers that you use to cluster them. So it is very common that people make this mistake. I even had somebody say, we did a bunch of transplant experiments to prove that these cells derive from those cells, but I wanted to see if it was right by running SPADE. I'm like, you did the experiment to see if it's right already. SPADE doesn't tell you anything except that they look the same in the markers that you've clustered. And you know, if you choose different markers to cluster, you will get different patterns. You know, these patterns are derived by what markers you chose to cluster on. Obviously, we tend to choose the things that are going to separate the data the best. But if you choose different things, you get different branches. If you choose a ton of myeloid markers, you get like 15 different myeloid populations. You get different myeloid arms. If you choose a ton of T cell markers, you only have like one myeloid arm, and you get like 10 different T cell populations. So it's just immunophenotypic relatedness. And so lastly, what can you do with this information? This is a different project that we're working on as part of a large collaborative focus. But you can see if you subdivide, these cells were in vitro cultured lymphoma cells treated with chemotherapy. But if you cluster cells by their cell cycle state, you'll find that a lot of things that you sort of think of as looking at across whole populations are actually quite different in different stages of the cell cycle. So here, looking at phosphorylated ATM, you can see that this picks up dramatically following chemotherapy treatment depending on what phase of the cell cycle you're in. So you can see much, much higher ATM activation in response to chemotherapy in the cycling cells as compared to the cells in G0. And you know, these differences are quite huge, and they're kind of masked when you're looking at the population as a whole. Similarly with H2AX, again, the damage is getting accumulated in these cycling cells, not in the cells that are in G0. And this, again, sort of supports like things like the stem cell hypothesis that these quiescent cells are not getting killed by chemotherapy because they're not in a cell cycle state to allow it. So I see Harris is here, so I'm going to wrap up. Just in conclusion, we've sort of developed and validated a methodology for using mass cytometry to assess a cell cycle state. This really only requires as few as 4 markers. And if you just want to do S phase, it only requires IDU, which you weren't using anyway. And so from that, you can do a system-wide analysis of proliferation across, in this case, normal and human hematopoiesis, but just about any kind of experimental system you might be wanting to do with the CyTOF. And so this can allow you to generate valuable information about hematologic malignancies and chemotherapy response. And you can combine this with lots of other variables of looking at response at the single cell level. And that's a great lead into Harris's work, because he's used this cell cycle to look at how cell cycle correlates with apoptotic signaling. But you can see how cell cycle correlates with all sorts of signalings, states that you might be interested in. So I just want to acknowledge the people in the lab that helped out a lot with this. Obviously, all the people on our lab team are fantastic, as you guys have probably gathered during the course, particularly Gary. Most of my work has been built off Sean and Aaron's work on the CyTOF and Matt Clutter's previous fluorescent flow cytometry cell cycle work. We get a lot of help from people like Angelica and Rachel in the lab. Michael Lindemann and Peng Kui were really instrumental in getting SPADE working, and then I have a variety of hematology clinical collaborators that help me get samples and design these experiments. So with that, I'll end. Thank you very much. Any questions?
Greg, back to your AML sample, you had 2 different phenotypes basically there. And when you, so you're going to use those as prognostic indicators for patient outcome.
Hopefully.
Hopefully. Can you go back and actually
Takes a lot of patience.
pull those, using something like flow, be able to pull those cells out and sort and see what kind of mutation it is?
Yeah. I mean, we spent a lot of time actually in doing this to make sure that we've titrated our markers just so, and we run the experiments just so, so that when we get these plots by mass cytometry that they correlate with plots that you get by flow cytometry. And so I've done a whole series of mouse experiments with one of my collaborators to like completely reproduce the gating that they've done on a mouse progenitor experiment that they've been doing. But it takes a little time to get things to line up exactly, but it can be done. I know Sean and Kara are doing work with B cells where they're describing patterns by cytoph and then going back by fluorescent cell sorting and then sorting out those populations, and they're getting the populations that they're seeing. So that's the best way to do those kinds of experiments is you define the immunophenotype that you're interested in, you define the biology, and then you narrow down based on being able to screen 20 markers or 30 markers. What are the 6 or 7 markers that I really need to sort this population? Then you do a separate fluorescent experiment to sort those cells out. So that's a feasible experiment. It just has to be done in 2 steps.
In the image stunning case, does the size or number of circles correspond to the number of cells?
Yeah, roughly, but Aaron will get into that more.